Morning there, friends. It's a beautiful morning, and it's always wonderful to have you here while we discuss science. Today, we're just going to look into the principles and application of biotechnology. We're talking about biotechnology. What is biotechnology? What does it entail? Now, we established in a former video that when you're talking about biotechnology, you're talking about the use of our knowledge of biological systems and computer, molecular engineering, molecular biology, genetics. The use of all this knowledge to manipulate genes in such a way that we bring out better outcomes for human beings. So, biotechnology has application in so many places, in the industry, vaccine production, antibiotics production, fermentation of substrates to make wine, alcohol, all these things are under biotechnology. When you're talking about biotechnology, you're talking about the totality of all of these things, the application of our knowledge used to manipulate genes in such a way that we have desirable outcomes. And so this is important to note and to understand. Now, when you understand what biotechnology is, the next thing to ask is what are the principles and tools that are used in biotechnology? There are quite a number of principles and tools that are used in biotechnology, and as a scientist, it's very important for us. Number one among the principles and tools being used is genetic engineering. When you hear genetic engineering, what should come to your mind? Genetic engineering refers to totality of the methods that are used to manipulate, play around with the sequences of genes, manipulate the sequences of genes in such a way that we begin to have desirable and better outcomes. This is quite important. You have many different techniques, like gene cloning, like recombinant DNA. These are all techniques we will discuss as we go forward. So all these techniques are used with the goal in mind that we're going to produce a better organism. We're going to produce an organism with better outcome. We're going to produce an organism or organisms that have, outcomes that are desirable. Or we're going to produce substrates, or we're going to produce products that are better because we have been able to manipulate genes. We have been able to change genes around in such a way that we have much better outcomes. So that's what we do in genetic engineering, and there is a lot under genetic engineering happens under genetic engineering. Now, apart from genetic engineering, we have what we call cell culture technology, which is another principle and another tool that we use to work in biotechnology. And when you're talking about cell culture, you realize that it is important for scientists to be able to culture cells. What do you mean by culture cells? To be able to grow cells. There are different types of cells. There are plant cells, there are animal cells, there are microbial cells. All these cells, it's important that scientists are able to, make many copies of these cells in the laboratory. So proliferate the cells and make many copies. Now, the question is, why do you want to be able to make many copies? You want to be able to make many copies because many times for cells, it's easier to understand them if you grow a population of such cells, not just a single cell. If you have 1,000 microbial cells, 1,000 human cells, 1,000 plant cells, 1,000 animal cells, whatever it is, you are able to study them better. You look at them under the microscope, you subject them to different conditions, and you're able to see the different outcomes that you have from varying the conditions, the temperature, pressure, acidity, alkalinity, all these different conditions. You can vary them and then look at the outcome. And what's the end goal? The end goal is to be able to produce products or organisms that are enhanced, that are better than what they were before. That's the general idea about technology. You're trying to produce products that are better than what you had before. So this is important to note. You have genetic engineering, you have cell culture technology. It's also important to know that in biotechnology, we make use of vectors. There are many different types of vectors. Now, many times in biotechnology, we're trying to create different products, we're trying to create new organisms. There may be need to transport plasmids, different genes, maybe small sequences of genes may need to be moved around from one organism to another. And when there's need to do this, sometimes there's need to use a vector. There are different types of vectors that can be used. You can have viruses being used as vectors. You can have organisms self being used as vectors. So just in order to move these genes around, because you want to move them from one place to another place, and then sometimes you want to proliferate, make many copies of the gene of interest. So you have a gene that you're interested in, just the way you have a car factory. You want to make many copies of that gene that you're interested in. So you want to put it inside an organism that proliferates really, really fast. And so over a short period of time, this organism has made many copies of itself. And these many copies of itself, you now use many copies. That means that your gene of interest, you've also made many copies of that gene. So this is quite important to note. Now, also in biotechnology, under biotechnology, we have fermentation technology. Fermentation is really important. There are a wide range of products that are made and consumed by humans that have fermentation at the base of the production of these products. So you have your alcohol, your wine, your so many different types of alcoholic beverages, undergo fermentation process. And in the fermentation process, what you have happening is that you have your organism of interest and you are using it to ferment different types of substrates, your barley, your hops, your rice, whatever it is, different types of substrate. And that way, when you tweak the condition, change the conditions around pressure, temperature, alkalinity, acidity, salinity, all these things, when you change them around, it gives you different outputs. So it's under biotechnology that we do all these varying experiments. I talked some time ago about brewing science, where I said you do all these experiments in order to see what are the outcomes from these experiments, which ones are desirable, which ones are undesirable. All of these are under biotechnology, and it's important to note. Also note that under biotechnology, we have enzyme technology. Enzymes are biocatalysts. And what do you mean by that? We are saying that enzymes are what we use to speed up the rate of biochemical reactions. So a reaction is occurring within the body, or we are doing it in the lab. We're doing it in the lab and we need to speed up that reaction. We can, use an enzyme to speed up. So when you say speed up, you mean that the rate at which the reaction will take place will be faster. It won't be slower because you have an enzyme. So enzymes are also there. There's enzyme technology. Biotechnology looks at enzymes, usefulness of enzymes, where they can be used, what reactions they can be used to work on. And this is in order to enhance, our products, to make our products much better. And a lot of enzymes are used in a lot of industrial processes. A lot of industrial processes make use of a lot of enzymes. Production of antibiotics, production of vaccines, production of different pharmaceuticals, nutraceuticals, all these things you want to make use of enzymes. So this is really important for us to know. Then it's important for us to know two more things before we go. Molecular diagnosis. A lot of diagnosis that we do when we use ELISA, we use PCR, and other very advanced biotech techniques. These are all under biotechnology. So we use all our knowledge of biotechnology, we use it in order to create ways and methods to use to do diagnosis, especially in the clinical science and the clinical area. So our knowledge is used in that sense, and then we create ways to be able to diagnose diseases very, very quickly with very high specificity. You use PCR, you use ELISA, you use different kinds of rapid diagnostic kits, RDTs, rapid diagnostic tests. A lot of these are the products of biotechnology. And this has helped to speeding up the rate at which science is able to identify different pathogens, and be able to help medicine to move forward very fast. Then lastly, we talk about bioinformatics. This is the area that combines our knowledge of biology with mathematics, statistics, and computer science. And this is important. We generate large amounts of data, we sift through this data, and we're able to, generate results and inferences that we can use to make human life much better. So when you have a convergence of biology, molecular biology, genetics, and computer science, statistics, and computing, all of this together to do very complex analysis that now, give us results, inferences, information from which we can draw inferences to make life better. That's the idea. To help to cure different types of illnesses and then make life better on the general. That's what we do in biotechnology. So I hope you've gotten a grasp of what we are talking about when we talk about the principles of biotechnology. We'll run another video this week where we talk about applications where we apply biotechnology in different areas of human life and existence. Thank you for listening to this video. Remember to like, share, comment, ask questions, talk to your friends about it. Let's grow our capacity in science. Let's grow our ability in science. Let's grow what we know in science so that we don't do science that is substandard. Thank you for listening.